Right now, moving through your body is a liquid organ made of 4 wildly different components and at least 8 major identity codes, and getting even one of them wrong during a transfusion can kill you. Type O blood is the blood type defined not by what it has, but by what it's missing. No A sugar molecules, no B sugar molecules sitting on the surface of its red blood cells. About 45% of people carry it, making it the most common blood type in many populations, though the exact split shifts depending on ancestry and geography. To understand why that absence matters, picture every red blood cell as a delivery van, and picture the immune system as a checkpoint guard scanning every van that rolls through. A and B antigens are like company logos painted on the side of the van. Flashy, specific, and instantly recognizable to a guard trained to spot them. A Type O van has no logo at all. It's just plain gray steel. Because there's nothing on it to flag as foreign, it can roll through almost any checkpoint in the body without triggering an alarm, which is exactly why Type O negative blood is called the universal donor and gets rushed to trauma bays when there's no time to test a patient's type. Unlike Type AB blood, which carries both logos and can therefore only donate to a narrow slice of the population, Type O's blankness is its superpower. The real-world anchor here is emergency medicine itself. Ambulances and battlefield medics stockpile O negative because it buys time when there's none to spare. However, that same blankness comes at a cost. People with Type O can only receive Type O blood themselves because their immune systems build antibodies against both A and B markers the moment they encounter them. Universal to give, but the pickiest of all when it comes to receiving. The ABO system itself was first mapped out by the Austrian physician Karl Landsteiner in 1900, when he noticed that mixing blood from different people sometimes caused it to clump, a discovery that turned transfusion from a lethal gamble into a science with rules, and one that eventually earned him a Nobel Prize. Type A blood. Type A is defined by a single sugar molecule called the A antigen, stitched onto the outer membrane of every red blood cell. Roughly 40% of the population carries some version of it, and the antigen itself is built by an enzyme that adds one specific sugar unit onto a base molecule already present on the cell surface. Think of red blood cell manufacturing like a factory assembly line, where a bare product moves down a conveyor belt and the stamping machine presses a company logo onto it before it ships out. In Type A cells, the stamping machine presses on the A logo, and every immune cell in the body grows up recognizing that stamp as home turf. Anything without it, or with a different stamp, gets treated as an intruder. This is different from Type B blood, where the stamping machine presses on a completely different sugar shape instead, meaning A and B cells look like products from 2 rival companies that don't recognize each other's branding. The real-world anchor is the transfusion chart taped to the wall of every blood bank. Type A can donate to A and AB, and receive from A and O. A set of rules doctors check in seconds before any transfusion, because a mismatch triggers a violent immune attack. However, that stamped-on logo is also a liability outside of transfusions. Some pathogens have evolved surface molecules that mimic the A antigen closely enough to slip past immune surveillance. One reason researchers have studied links between blood type and susceptibility to certain infections. The logo that protects you in the blood bank can be the very thing a clever invader learns to copy. Type B blood. Type B carries the mirror image sibling of the A antigen, a different sugar molecule attached by a slightly different version of the same stamping enzyme. It's less common globally than A or O, though it appears at notably higher frequency in parts of Central and South Asia, a pattern population geneticists have traced through migration and disease pressure history over thousands of years. If Type A cells are stamped with one company logo, Type B cells are stamped with a rival logo, built from the same manufacturing line, but with one different ingredient swapped into the recipe. Close enough that the underlying machinery is identical, different enough that the immune system treats them as total strangers. That single ingredient swap is controlled by one gene with multiple versions called alleles competing for the same slot on chromosome 9. Compared to Type AB blood, which manages to run both stamping programs simultaneously, Type B runs only its own, meaning a Type B person's immune system stands ready to attack any A-stamped cell it encounters. The real-world anchor is global blood supply logistics. Because B is rarer in some donor pools than the demand from B patients requires, blood banks in certain regions actively recruit B-type donors to keep supply balanced. However, this rarity becomes a genuine emergency medicine problem. A hospital that runs short on B-negative blood in an area where few donors carry it has far fewer backup options than one running short on the far more common O-type. Rare isn't just a statistic here. It's a supply chain risk written into human veins. Type AB blood. Type AB is the rarest of the four major ABO types, found in only a small slice of the population, and it's rare precisely because it requires inheriting both the A version and the B version of the antigen gene from two separate parents. Picture the red blood cell factory again, but this time the assembly line has two working stamping machines instead of one, pressing both the A logo and the B logo onto the same cell without conflict. Because an AB immune system grew up surrounded by both logos from birth, it never learned to treat either one as foreign. It recognizes both as home turf. That single fact makes type AB the universal plasma donor and universal red cell recipient, sitting at the exact opposite end of the spectrum from type O, which recognizes neither logo and therefore attacks both. Doctors call this the receiving mirror image of the donating relationship, where O gives to everyone but takes from almost no one, AB takes from everyone but gives red cells to almost no one outside its own type. The real-world anchor is neonatal and surgical medicine, where AB plasma, the liquid portion of the blood, is prized because it won't attack a recipient's own A or B cells, making it useful in emergency plasma transfusions regardless of the patient's type. However, because AB is so rare, supply is chronically thin, and blood banks often have to ration it carefully for the specific surgeries and trauma cases where a universal plasma donor actually changes the outcome. The type built to get along with everyone is also the hardest one to find when you need it. The Rh factor. The Rh factor is a separate protein system layered on top of the ABO type, named after the rhesus monkeys in which it was first identified back in the 1940s by researchers including Carl Landsteiner and Alexander Wiener. About 85% of people carry the main Rh protein, called RhD, on their red blood cells and are labeled positive, while the remainder lack it entirely and are labeled negative. If ABO antigens are company logos, the RhD protein works more like a keycard sitting in a scanner slot on the cell's surface. Either the keycard is physically present, granting a certain kind of access, or the slot sits empty. Unlike the ABO system, where antibodies against the missing type exist naturally from early childhood, the body doesn't automatically build antibodies against RhD. It only starts manufacturing them after direct exposure to Rh-positive blood, the same way a security system only flags a specific keycard as fraudulent after seeing it used once. This is why Rh combines with ABO to create the familiar 8-type system doctors reference on donor cards, from O negative to AB positive. The real-world anchor is pregnancy. If an Rh-negative mother carries an Rh-positive baby, her immune system can build antibodies against the baby's Rh protein after exposure to its blood, which is why Rh-negative mothers routinely receive a preventive treatment called RhoGAM during pregnancy. However, before that treatment existed, this exact mismatch caused a serious condition in newborns, showing how a single missing protein can turn a mother's own immune system against her child. One keycard, absent or present, rewrites the rules of an entire pregnancy. Plasma is the pale yellow liquid that red and white blood cells and platelets float in, and it makes up around 55% of total blood volume, roughly 90% of it just water. Dissolved inside that water is a cocktail of proteins, salts, hormones, clotting factors, and nutrients being ferried from one part of the body to another. Picture plasma as a river system running through a city, with barges constantly loading and unloading cargo at every dock. Nutrients get picked up at the intestines, waste gets dropped off at the kidneys, hormones get released from glands and delivered to distant target organs, all riding the same current. Unlike red blood cells, which are built almost entirely for a single job, Plasma is a multitasking transport medium carrying dozens of different cargo types simultaneously, including the antibodies your immune system produces and the clotting proteins that platelets rely on to finish sealing a wound. One of its most important passengers is a protein called albumin, which keeps water balanced between blood vessels and surrounding tissue, preventing the kind of swelling that shows up when liver disease or severe malnutrition drives albumin levels down. The real-world anchor is plasma donation itself, a process called plasmapheresis, where the liquid is separated out and the red cells are returned to the donor, allowing plasma to be donated far more frequently than whole blood. However, because plasma carries so many different functional proteins at once, a shortage of even one of them, a single missing clotting factor for instance, can cause serious problems even while every other component of the blood functions perfectly. The river carries everything the body needs, which means losing even one cargo type can flood the whole system with consequences. Plasma also carries the body's chemical messaging system, ferrying hormones like insulin from the pancreas to muscle and fat tissue across the body, and buffering compounds that keep blood's acidity locked within an extremely narrow range. A range so tight that even small deviations can disrupt how enzymes throughout the body function. Red blood cells. Red blood cells, called erythrocytes, are the oxygen delivery workhorses of the bloodstream, and there are roughly 25 trillion of them in an adult body at any given moment, each one living for about 120 days before being recycled. Picture a fleet of delivery trucks with no cargo hold at all. Instead, every square inch of the truck's exterior is covered in hook-shaped molecules called hemoglobin, and oxygen molecules simply latch onto those hooks as the truck passes through the lungs, then release again wherever oxygen levels run low. That hemoglobin is also what gives blood its color, turning bright red when saturated with oxygen and a darker shade when it isn't. Uniquely among the body's cells, mature red blood cells eject their own nucleus during development, sacrificing the ability to repair or reproduce themselves in exchange for more internal space to pack in hemoglobin, a trade no other major blood type segment makes. That's a sharp contrast with white blood cells, which keep their full nucleus and genetic machinery because their job depends on active decision-making, not raw cargo space. The real-world anchor is anemia, a condition where either the number of red blood cells or the hemoglobin inside them drops too low, leaving tissues starved for oxygen and causing the fatigue and paleness anemia is known for. However, this design has a built-in vulnerability. Because mature red blood cells can't repair internal damage or divide to replace themselves, any defect baked into them at birth, like the misshapen cells of sickle cell disease, stays with that cell for its entire short lifespan. A truck that can carry more cargo than almost any other cell in the body, built by giving up the one thing it would need to fix itself. New red blood cells are manufactured continuously inside the spongy bone marrow of the ribs, spine, and hips, in a process called erythropoiesis that's regulated by a hormone called erythropoietin, released by the kidneys whenever they detect falling oxygen levels. The same hormone that has been abused by endurance athletes seeking an artificial boost in oxygen-carrying capacity. White blood cells. White blood cells, called leukocytes, are the immune system's active defense force, and although they make up less than 1% of total blood volume, that thin sliver includes several distinct cell types, each trained for a different kind of threat. Picture a military garrison stationed throughout the body, with different units assigned to different jobs. Some patrol constantly, some rush to the scene of an injury within minutes, and some remember old enemies for decades after the first encounter. Neutrophils act like first responders, arriving fastest at a site of infection to swallow and destroy invading bacteria in a process called phagocytosis. Lymphocytes, including B cells and T cells, function more like an intelligence division, recognizing specific pathogens and either producing targeted antibodies or directly killing infected cells. And it's this same lymphocyte memory that makes vaccines work years after the original shot. Unlike red blood cells, which are locked into a single, unchanging job for their entire lifespan, white blood cells can squeeze out of blood vessels entirely and crawl through tissue toward an infection site, something no red blood cell is built to do. The real-world anchor is the common blood test called a white cell count, which doctors use as one of the first signs of infection, since counts spike when the garrison mobilizes against an invader. However, this defense force can turn catastrophic when it malfunctions. Leukemia occurs when white blood cell production spirals out of control, flooding the bloodstream with immature cells that crowd out healthy ones and can't do their job properly. The same army built to protect every cell in the body can, when its own command breaks down, become the threat it was built to fight. Beyond neutrophils and lymphocytes, this garrison also includes monocytes, which mature into cleanup specialists called macrophages that engulf dead cells and debris left behind after an infection, and eosinophils and basophils, smaller specialist units that respond to parasites and drive the inflammation behind allergic reactions. Platelets, also called thrombocytes, aren't full cells at all. They're small fragments budded off from a much larger parent cell in the bone marrow called a megakaryocyte, and a healthy body maintains somewhere between 150,000 and 450,000 of them in every microliter of blood. Picture a construction crew on permanent standby, tools already loaded, waiting for a call that a section of road has cracked open. The moment a blood vessel wall is damaged, platelets rush to the site and change shape, growing sticky spiked arms that let them clump together and plug the breach within seconds, a process called primary hemostasis. That plug is only step one. Platelets then release chemical signals that summon plasma's clotting factors to build a tougher, more permanent mesh made of a protein called fibrin, reinforcing the patch the way a construction crew pours concrete over an initial emergency fix. Unlike white blood cells, which travel through tissue hunting for threats, platelets stay confined to the bloodstream and only activate when they physically detect a break in the vessel wall itself. The real-world anchor is bruising and clotting disorders. A low platelet count, called thrombocytopenia, leaves people prone to excessive bleeding from even minor injuries, while conditions like hemophilia involve a breakdown further down the clotting chain rather than the platelets themselves. However, the same clumping response that saves your life during an injury becomes dangerous when it triggers inappropriately inside an intact vessel, forming clots that can block blood flow to the heart or brain. A repair crew fast enough to save your life in seconds, and, in the wrong circumstances, fast enough to end it just as quickly. Bombay blood group. The Bombay blood group. The Bombay blood group is one of the rarest blood types on Earth, found in roughly 1 out of every 10,000 people in India and far fewer elsewhere, first identified in Mumbai in 1952. To understand it, you first need to understand that both the A and B antigens are built on top of a foundational base molecule called the H antigen. Without that base, neither logo can be stamped on at all. Think of it like a locksmith who's run out of blanks. It doesn't matter how skilled the stamping machine is at pressing the A or B pattern if there's no base key to press it onto in the first place. People with Bombay blood type lack the H antigen entirely due to a mutation in the gene that builds it, which means even if they've inherited the genetic instructions for A or B, those instructions have nothing to attach to. This makes Bombay blood type incompatible with every other blood type, including O, because O blood still carries the H antigen, just without A or B stamped on top of it, and a Bombay immune system will attack that H antigen as foreign. Unlike type O, which is the universal donor precisely because of its blank surface, Bombay blood can only be safely given to and received from other Bombay type individuals, making it far more restrictive than any of the 4 main ABO types. The real-world anchor is the serious logistical challenge it creates in emergencies, since patients with this type often have to rely on rare donor registries or even banked units of their own blood collected in advance. However, because it's so easily mistaken for type O on standard tests, misdiagnosis has historically led to dangerous transfusion reactions when doctors weren't specifically screening for it. The type so rare it can be hiding in plain sight, mistaken for the most common blood type of all. The mutation behind it disables the gene responsible for building the H antigen in the first place. And because it's a recessive trait, both parents typically have to silently carry a single copy of it for a child to end up with the full Bombay phenotype, which is part of why it clusters in populations with a history of marriages between close relatives. Cord blood. Cord blood is the blood left inside the umbilical cord and placenta after birth, and it's remarkable not for what type it carries, but for what's floating inside it. A rich concentration of hematopoietic stem cells, the parent cells capable of becoming any type of blood cell in the body. Picture a seed bank, sealed away at the exact moment of collection, packed with cells still holding the full instruction set to become red blood cells, white blood cells, or platelets, rather than being locked into one specialized role already. Bone marrow contains these same stem cells in adults, but cord blood stem cells are notably more adaptable and less likely to trigger immune rejection, since they haven't yet been exposed to the wide range of pathogens and immune experiences that shape an adult cells over a lifetime. That relative immune naivety is what makes cord blood transplants more forgiving of imperfect donor matches compared to adult bone marrow transplants, which usually demand a much closer genetic match between donor and recipient. Unlike ordinary donated blood, which gets used almost immediately, cord blood is typically frozen and banked for years, preserved in liquid nitrogen until it's needed for a transplant. The real-world anchor is the treatment of blood cancers like leukemia and certain inherited immune disorders, where a cord blood transplant can effectively rebuild a patient's entire blood-forming system from scratch. However, each cord blood collection yields a strictly limited volume, which means the stem cell dose is sometimes too small to treat a full-grown adult effectively, restricting its use in certain cases to children or smaller patients. A single sealed vault of biological potential collected once at the very beginning of a life and then held in reserve for a crisis decades later. Synthetic blood. It is, more accurately called a blood substitute or artificial oxygen carrier, is an engineered fluid designed to perform some of blood's core functions without needing a human donor at all. Rather than being grown, it's manufactured, using one of two main approaches. Hemoglobin-based oxygen carriers that use purified or lab-produced hemoglobin, or perfluorocarbon-based fluids that can dissolve unusually large amounts of oxygen directly into a synthetic liquid. Picture a 3D printer built to manufacture a rough approximation of a delivery truck fleet. It can't replicate every feature of the original vehicle, but it can nail the one function that matters most in an emergency: hauling oxygen from one place to another. Unlike red blood cells, which carry hemoglobin safely packaged inside a protective membrane, early hemoglobin-based substitutes used free-floating hemoglobin, which turned out to scavenge a signaling molecule called nitric oxide from blood vessels, causing dangerous constriction. A side effect researchers have spent decades working to engineer around. The appeal is enormous. Synthetic blood doesn't require typing or cross-matching the way donated blood does, since it carries no ABO or Rh antigens to trigger immune rejection. And it can theoretically be stored far longer than the roughly 6 weeks donated red blood cells last on a shelf. The real-world anchor is military and disaster medicine, where a shelf-stable, universally compatible oxygen carrier could be a genuine lifesaver in situations too remote or too urgent for a standard blood supply chain to reach in time. However, no synthetic blood product has yet matched the full complexity of real blood. None of them clot, fight infection, or regulate the body's chemistry the way genuine plasma, platelets, and white cells do. So every version so far is a stopgap, not a replacement. Science can now manufacture blood's most famous function in a lab, but it still can't manufacture blood itself. In 2022, researchers in the United Kingdom successfully transfused lab-grown red blood cells, cultured from donor stem cells rather than assembled synthetically, into human volunteers for the first time. A step that sits somewhere between traditional donation and true synthetic manufacturing, and hints at where the next generation of blood substitutes may come from. 4 antigen systems, 5 cell types, and 1 rare mutation that can erase them all. Together, they form a single transport network where every piece depends on the others to keep the body alive. And if a video this deep into your own bloodstream got you leaning in, subscribe for the next one.